Phospholipids are very important molecules for the study of biology because the phospholipids, these are cell components, and these make up the cell membranes. This is going to be any cell membrane, whether it is a cell membrane around the outside of the cell, whether it's one inside the cell, whether it's found in a human cell, bacterial cell. All cell membranes are going to be made up of these phospholipids. So if we talk about the structure of a phospholipid to start with, phospholipids are really constructed from a few different types of molecules. We are going to have one glycerol molecule. And so I'm going to draw that glycerol right here. Glycerol is a little 3-carbon alcohol. So it's got 3 carbons, and each one of these carbons has a hydroxyl group coming off of it. And then we can fill in with our hydrogens to make sure that every carbon gets 4 bonds. And so we're going to take that, and now in 2 of these positions, so if we'll say right here and right here, we're going to end up with a fatty acid attached. So fatty acids get attached in both of these positions. So we're having one glycerol, we're going to have 2 fatty acids attached by dehydration reactions in each of those positions. And then the other component that we're going to get is we're going to get one phosphate group. And the phosphate group is going to get attached at the 3rd position up here. So I'm going to write that in like this. So once all of this is put together, we will have that structure called the phospholipid. Here we have a look at how this is all going to fit together. So right here, we can see our glycerol backbone. Okay, so that was where the glycerol was at. What's shaded in blue here, that was the 2 fatty acid tails. Now notice that we can have fatty acid tails that are different from each other. In this example, we've got one that's saturated up here, and we know that it's saturated because we do not see any double bonds inside here. And then this one is going to be our unsaturated. And then over here, we have that phosphate group that we talked about. And sometimes, many times, you do have something else attached to the phosphate group. So we do see that here, and a lot of times that does contain some nitrogen inside of it. Now, looking at this, when we get this total structure together, notice that this molecule, it does have this very large portion over here, which is all going to be, mostly it's hydrocarbon tails. So this part is very hydrophobic. And it's hydrophobic because they have lots of non-polar covalent bonds there. But then when we look over here at what's shaded in green, this has lots of polar covalent bonds. And the reason that we have lots of polar covalent bonds is because there's so much oxygen and there's so much nitrogen in there. And so we end up with this molecule that really has 2 different sides to it. We have one side that is pretty polar or hydrophilic. It likes to interact with water. And that's what we tend to call the head of the molecule, or the head group. And then we have the part that's shown in blue, which we tend to call the tails. And that's the hydrophobic non-polar part. When we have membranes formed, the way that it works is that you can see in this image here that we'll have a bilayer. And it's a bilayer because we're going to have 2 layers of phospholipids. And these 2 layers will face each other so that all of those tails are on the inside of the membrane, and all of those polar heads or head groups are on the outside of the membrane. And this is going to work very well for the cell because these will hold together just like this, because it's just going to be very energetically favorable, because it puts all of the polar stuff next to other polar stuff, which makes them happy. And all of the hydrophobic non-polar stuff gets to hang out together, which makes them happy. And also, if we're talking about inside the cell or we're talking about outside the cell, these are probably going to be water-based environments. And polar stuff is happy being next to water. So that will keep the polar stuff next to the water and the non-polar stuff away from the water, since non-polar is going to be hydrophobic.